And yes, I emphasized or we emphasized the data, and it's my great pleasure to introduce. It's a special webinar. We have two speakers, two young men from France, Benoit Petit-Demoulière and Damien Huzard. Benoit is the head of phenotyping department of FEN ICS, formerly known as Institut Clinique de la Souris in Strasbourg. And he's a key member of the French national infrastructure, Selfedia, and has extensive expertise in preclinical research and in vivo models. Damien, quite recently, a year or two ago, defended his PhD from EPFL in Switzerland. He's a behavioral neuroscientist and the founder of Neuronautics company, creator of metadata app, and his background is deeply rooted in the study of behavioral adaptation, stress, and home cage monitoring technologies. But when they came together, now I don't know exactly when, but maybe they met during the tea time. So they recently published a paper about data care, that is the new frontier of animal welfare. And without a further delay, I give the screen over to Damien and Benoit, please.
Thank you very much. So I will start the presentation, and first of all, I want to thank Vootele and also the CITE consortium and everyone that is from CITE. And indeed, I met Benoit and thanks to CITE, and I think it was like a really luck for me to be part of this doing one of my postdocs, because it really showed me what a good scientific network is about. And since I'm an independent scientist, I'm more scientifically productive thanks to the network I made with CITE than I was when I was stuck in my postdoc project. So thank you very much, everyone. And today we are really happy to talk about data care. So two parts of the talk today. I will start with some theory about metadata management, and then Benoit will continue. And after that, we will do two short demo about the tools that we are developing in order to improve data and metadata management in preclinical research. So what I want you to do now is to imagine that you're entering in a library, but you're just facing a pile of books. You were going there to find some information, you know is findable somehow, but because it's not organized, you cannot actually access and find the data that you're looking for. So we fixed this problem in a library by using some metadata, contextual informations that are making some sense about the author, the date, some unique identifications like the ISBN, and we are able to build shelves. And at the end, we organize the books, and tomorrow you go to the library, you find the book, you find the page that is interesting, and you have the information you were looking for. For this, context is king. Without the context, so the metadata that is actually representing the data that is included in the book, it would not have been possible. So, in short, the metadata is the data about the data. For example, in a preclinical research setting, the data could be the scientific results that you buried deep into a scientific paper. The metadata is all the context, all the information that would allow a reader or anyone else accessing your paper to actually find and give some sense, some contextual sense about the scientific result that you produced. So to do that, we need some guidelines, some constraints, and at the end, in preclinical research, what we want to do is to build some sort of shelf that allows us to find the important information about the studies, the research, the results that have been published in preclinical research. So there is the image disappearing here now, but imagine the mice are still here. And we have the contextual information that we could start gathering pretty easily on all our protocols, preclinical research about the mice. And then we organize them. We use those metadata in a software, and we can build this shelf. And it allows reusability of data. It's here, sorry. So 10 years ago, exactly 10 years ago, the FAIR Principle Guidelines article was published. And this was the paving way of what we are trying to build now. So the FAIR Principle, they stand for data that are findable, accessible, interoperable, and reusable. So something that is findable is that it should not take ages for someone to actually be able to find some specific data, and it has to be accessed easily. So there are still some rules to be respected, but the access should be feasible. The dataset should be able to interact, or the software between each other. And at the end, the goal is that anyone could be able to reuse properly some specific data. So now a bit more details about what really those four pillars stand for. So the findability is the fact that in a dataset, we should use unique identification, unique identifiers. And for example, an article has a DOI, a human can have an ORCID. We all have unique identifiers, and each time you're publishing something, you just attach your ORCID, and the identification problem is solved. We can do that for a lot of parameters in preclinical research using unique identifications always. The accessibility is then pretty easy. We are using computer, we are going on browser, we are using the HTTP or HTTPS protocols. Those are open, those are free. We can share data using those protocols. We should not share data with private and proprietary protocols like WhatsApp or Zoom, even though they're really effective in some specific situations. We should share data using some free and open protocols. The interoperability is going a bit more in details about the way the data and the vocabularies and the entities are organized within a dataset. This is to be done by following also some vocabularies that have been also made following the FAIR principles. And this is most of the time using the World Wide Web, so the W3C consortium that are paving the way with a lot of formats, a lot of rules that allow to define proper languages. And this is to be sure that when you're sharing a dataset, another machine can understand it perfectly. Another human as well, but here the machine is really important because it can navigate into the dataset much more quickly than we can. And the reusability is the fact that when you publish a data, you should put some attributes, some licensing. Who can reuse your dataset? Who can access it? Can they make money out of it? You have to be able to define and standardize the way you're sharing your data with the licensing. You have to justify the provenance, and this can already be done really easily with some sort of README documentation that are attached to project. It's like a pretty easy way to start. It's like free text description of what your data is describing, what has been done, and it's already better than nothing. Now we wanted to link this with the 3R principle. So you're all familiar with those, the fact that we have, when we're doing preclinical research, find ways to replace, reduce, and refine the protocols, everything that we do to the animals. But at some point, we realized that taking care properly of your data is actually taking care of the animal. So this is when Benoit came with this really nice name, the WELLFAIR framework. And the WELLFAIR framework that we published recently in Neuroscience Applied is literally the proof that data welfare is, by definition, animal welfare. So what we suggest in the paper is that by following this small timeline, like with tools and software that are taking into account the different phases between planning, acquisition, federation of data, and the extraction of the outputs, we can build tools that are fair by design or born fair, that allow everything that is going to take care in the research to be shared and following the FAIR principle, sorry. We also were lucky to be part of this really nice book about home cage monitoring, and we contributed in the data sharing and metadata chapter. And in this chapter, we were also like asking ourselves, how do we map actually the FAIR principles into the 3R principles? And we found that between the FAIR principles, we can define some welfare decisions that are directly related to the 3Rs. and that all of the sub-items of the FAIR principles are mapping to 2 or 3 of those welfare decisions. So with this kind of illustration, we can justify the fact that fair data and fair data management in the research facility is a way in the long term to follow the 3 Rs. So the take-home message here is really like to also make scientists realize that if the data set that they are generating are not usable, they are actually in a way wasting animal lives. And I think this is something that I want to emphasize and that people have to be aware more and more. So now I will give time to Benoit to connect.
And that should be okay. You should see my slide now.
Yes, perfect.
Yes, we can.
Yes. So thanks a lot, Vootele and Damien, for this great starting. And thanks to have showcased so well the welfare that we are now trying to share with you and to explain what's the behind and the moral contract that we have with the animals that we're using for animal experimentation. I think that's really something that is in the actual way of doing lab science and using animals. We really need to focus on the use of each animal in the building of this knowledge and the science. And the more and more we are considering that science is data. It's not only to have the papers and the articles, but it's what do we need to create a knowledge that will be helpful to build like medicines and drugs, but also to have the global knowledge of the biology in our case. It can be like creating virtual control groups or creating even virtual mouse to avoid to use the animals in the best way to have the replace of the 3R. But the question is, how do we consider to create this knowledge? And initially, when I was like a PhD, I was considering that knowledge was like a wall of brick that we were building on the previous layers of bricks of knowledge, and that we were just like piling up stones and stones of knowledge, and we would have trust of what was been published before, and that was the way I was like considering the whole thing. But the question is, do we really have all the bricks that help to create this knowledge? And now I think I moved to something that is quite a different point of view, because this creating on the pile of knowledge seems to be fragile at the end. And we hit this wall with the reproducibility crisis. That was something that appeared in the latest year, showing that, it's not so easy to reproduce published results in another lab for multiple reasons. But one of this is the lack of metadata that explain how to deal with. It can be protocol, it can be environmental conditions, it can be multiple things. But at the end, we are facing this wall. And the more and more I consider that creating the knowledge is more like a patchwork of knowledge that we are all trying to stitch together to create like a blanket that will be comfortable. And in this case, each of these parts of the patchwork will be like the data that you are able to grab. And currently, the publication are providing some data through the supplementary data, but that's quite poor in terms of the number of experiments that are done on animals compared to the ones that are really available in data in the publication. It's only a subpart, and even then it's quite not so easy to reach. And then there are also the data that are published in the unstructured repository. For example, in France, we've got the research-data-gouv.fr, which is like a data lake where you can put the data, but it's not labeled, it's not organized, it's just like shared but not open data. And there are also people that are doing a lot of putting a lot of data in collaborative libraries like the Zotero, because that allows them to share things with DOI. But at the end, it's not so easy to dig out and to find this data. And here I've got some multiple patches, but I can't create a blanket because there are plenty of missing data. And all this data is because all the data that is currently shared is only a part of the story, and they are not fair at all. So there is one exception that I wanted to highlight, because not only because I was part of it, but also because I really think that in the preclinical field, we should take a lot of info and insight from the IMPC consortium. So The IMPC consortium, for the ones that don't know that, it's multiple core facilities that were organizing themselves all around the world after multiple iterations and consortium to define metadata, to define protocols. They started all these facilities to create knockout lines for each of the coding genes of the mouse genomes. And the idea would be to have a standardized protocol and pipeline of tests and assays and metadata, and to share that freely for the community. So if you go to the website, you can type in the gene and you will see that there are multiple protocols, data, and metadata. And frankly, when you see the amount of work that it took to create this kind of data with the actual metadata and the curation and these kind of things, you see how big it is and how hard it is for a scientist to reach this level of quality. So take time to explore. It's completely free available. You just type in the website and you can check that. That's for me the only real concrete example of what could be a nice way where we should go. But the constraint is the IMPC website is only for the IMPC pipeline. So there is a defined pipeline, defined conditions, and not all the scientific community can just throw data in it. So we have to move forward with this in mind of what could be one of the example of sharing data. Because at the end, we've got these multiple pieces that are missing in our blanket, in the patchwork. And what is missing and not available is what we call the shadow data, because we are shaping the knowledge based on what we've got, not what we miss. When we are scientists, we are just taking all the clues that we've got and we try to make hypotheses and fill the gap. But at the end, if plenty of people try to create a solution to a protocol to find the data in the gap, and they failed that, because maybe the question was badly raised or maybe the protocol was not good. But at the end, multiple people will try to stitch the same pieces and will fail, and they won't share that because it's a negative result. So the question is, where are these missing pieces? And there are the things that are not collected, like the protocol did not capture that. It can be metadata, but it can be also data. It was collected but not published. So in this case, it's often the negative results. And otherwise, it exists, but it's not accessible to the other scientists. So either a bad design of the protocol, not sharing the negative data, that's something that is really a shame currently, because it's actually the bias of the publication, and the FAIR by design that is not accessible. So it's the A of the FAIR. And there are some examples of how that creates bias. So for example, there is this I love the survivorship bias that is quite common, but for the people that don't know, it's how do we make to create more robust planes during the war? And they were analyzing the bullet holes in the coming back planes that were the question is, all these planes had holes due to the bullets, and how do we reinforce the planes? And the thing is that we observe only the aircraft that made it back. And so what was visible on these planes was the part that where the planes could be hit and still they could survive. But the missing observation will be the most informative part, because at the end, the ones where you see there are no holes at all, that's the plane that didn't came back. And this one didn't survive. So that's the part that at the end was the more susceptible to be reinforced. So what we see is not the full phenomenon, but a subset that passed through a selection filter. And that's exactly what we are currently using as the data. The published data are the things that went through the whole selection filter. So we had a question and hypothesis, we raise the question, we make a protocol, we've got the data, and we publish it because it's exactly in line with the expectation. But all the other ones, all the failures, are not currently available. And it's not noise, it's the structure of the knowledge. I went to this direction and it failed. And it failed because I don't know, but at least it failed. And all the other people should have this knowledge, because that means that all the potential pathways through the knowledge have to be explored. And the less we do the same, the better it is for the animals and also for the progress of the science. So There are some efforts currently trying to promote the publication of the negative results. One in France, for example, I am not aware of other these kind of things, but in France, the ST3R created short notes, and this is to publish small papers. It's 2,000 words, something like that, but just to showcase something, even if it's negative, but it's just they are in this promotion of sharing the knowledge. And they are creating DOI, so it will be available to the community and this kind of thing. Because at the end, with all these missing parts, we are creating a lot of bias in the preclinical research. Sex bias, because there are multiple disease models where we are not using all the animals. There is a cost action about that that is currently starting. Sex as a biological variable, if you are interested in it. The thing is that, like I mentioned before, the positive results are more likely to be reported than the negative ones. And a lot of metadata are currently never shared at all. It can be the regions, the protocol, and even the raw data before the transformation and the stats. So all the respect for the source and the science requires to share all these data points. Okay, but how do we share these data points? We are currently building a tool that is called FAIR3R, and that's exactly to fulfill this gap. I mentioned before the IMPC, which is a website that was built with a lot of efforts on standardizing the thing. FAIR3R is not the opposite, but it's offering a lot of flexibility to the people that will put the data in it. So the idea is that this tool will be on the server. Currently, we've got one server in Strasbourg Uni for the FAIR3R.fr, but basically it will be open sourced, and every institute or country will be able to deploy its FAIR3R instance. And the goal is to have a portal where the scientist will be able to drop the data and get a DOI, and will be referenced with all the available information and metadata to refer to the science and to make it findable. So we are using CCAN as the backend. CCAN is a worldwide used open data portal. It's like the data.gov in US is using this kind of tool. So it's really a massive tool, easy to follow with a lot of developers. So in terms of safety and this kind of thing is really great. And we are just customizing it to allow for all purposes. All the plugins will be open source. And the FAIR repository will have only one source of truth. Previously, Damien showed you that, for example, for the scientist, you could have an ORC ID as a unique identifier, sorry. And that's the same for plenty of things. We can have unique source of truth for plenty of knowledge. And we are creating a form that will be synchronized among all the instances of FAIR3R, and that will provide a good way to label the project. And also, all the communities will be allowed to add their source in this form, because, for example, we are currently organizing the things on the mouse and the rats because we have this knowledge. But if the community of the Xenop, if interested in putting data, they may have their own repository of like all the potential strain in the Xenop. I don't know if that happens, but that maybe. I'm not really an expert. So and they could plug that source of knowledge in the global form. And I've got a video. I hope it will go. It was going just well before. So that's a live demo. I didn't want to make it live. So you've got an organization you are a member of, and there is a form that allows you to create a data set in the form. Oh, it's quite ugly. I think I maybe. Oh, yeah. Hopefully the connection will go. Yeah. So I'm just putting a title, a description. That's the project I want to share with the community. After that, there are multiple things that you can fill in. The license is pre-filled. Organization is your organization. You can define it as a private repo or public until you want to share it with the people inside your team, or after that to release that to the public. And then you've got the publisher name. Currently, basically, it's where the instance lays. So as we are building it, it's currently our institute. It was, when I recorded it, it was Institut Clinique de la Souris. And then there are all the people that are in the project. So you can type in your first name, last name, and then the ORC ID is automatically finding you because it was in the source of truth. The form is looking for the ORC ID. Same for the ROR. It's if you've got an institution that is entered that has the ROR, it also allows you to select the project, the credit, like what did you make in the project using the guidelines from the credit guys, and you can add multiple contributors. Then you can select the model. So for example, if you select the mouse, that opens a specific form dedicated for the mouse. And then you can type in the strain, and that will look in the repo where we've got good knowledge, and that will tag them. And then you can select what are the things that you did on this model and the data that you will publish. So I checked all the ones to showcase. And for example, there was a genetic modification of this gene. I type in by just to show. And when you are entering the gene, you are showing also all the data that were directly crawled, like the NCBI, the MGI, the Ansible IDs. All the data were directly taken from the source of truth because that's the repo that we're using. And then you can time also the mutation. That's also something that is digged through these kind of things. Same for the molecules. We are directly going from the chemistry EBI, and some like small global things about what is the treatment, the protocol, the design, these kind of things. That just two typing free text, but that will be all this info. So the gene line, the name, these kind of things, the protocol, etc., will be attached to this description and to the DOI, which means that all the people that will be looking for MGI ID, for a gene name, for some protocol with specific diet of this specific molecule, they will be able to find your data because it's labeled with all this controlled vocabulary that is defined by the community. So it can be also disease model, so it's using the DOID. If there are additional models that will be of interest, that's also something that could be plugged to this form. It's live built with the form that we are creating. Also the organ of interest, this kind of thing. So once you are labeling the whole project, you can then add the data, and then that will be an upload of what you want. It can be directly a file, a CSV, an XLS, anyway. It can be also a link to another resource, like you uploaded your big data in Zotero or on the live folder, or you can also create directly a table and enter the number if you are really on the geeky side. But anyway, I would just finish this part just to showcase how we are creating this project. And then you see that it's creating this directly with a DOI that is quotable and that will be findable with the description, with all the metadata that you entered and that are using a controlled vocabulary. So the idea is really to aim for maximal findability and also to design it to be flexible. And then once you've got the data in it, I just made a second video. If you are creating a project with multiple data sets, so that's an IMPC project. I take it as an example. I uploaded a CSV, and directly it's crunching the data and putting it in a data table that you can live explore. That's great because that means that AI tools will be able to explore all the data sets that you are making publicly available. And in this data, there will be not only the data by itself, and when YouTube will give me the ball, yeah, okay, but there will be also the metadata, like the equipment model, this kind of things. So that means that with this available, I'm opening to AI tools, for example, all the data, but linked to the metadata that are available in the data sets. And that's there are also some things of in additions like doing live. Sorry for the lagging. Yeah, you can also live plot the data if you want that you just upload to make so the people that will visit the website and find the data sets will directly be able to have a live exploration of the data to see, okay, what does it mean? So you can build whatever you want with multiple kind of graphs, like I'm dropping the genotypes and I'm trying to see what the grip means. And that's the values of all the animals that were made. But I know that there are multiple genders, so the sex is there. And so I've got the males and the females and this kind of thing. So that's not to be said, and that won't be as efficient as the IMPC portal, which is doing statistics of really diet exploration. That's just to have a live vision of what you are currently dealing with in terms of data, etc. And that's all for me. So I will either switch back or you take the ball, Damien, and I scroll the slide.
Actually, I can share. Yeah.
Okay. So I unshare my.
Okay, sorry for a new transition. Okay, perfect. Yeah. So now, quickly, I will mention how we move from FAIR data to AI and potentially to virtual control groups. So the idea is that we can see different building steps that allows us to go from standardization to actually making sense and virtualizing the animals. So first, and that's where we are actually working on, is the standardization, defining some standards, proper ways to incorporate all the metadata, the sensors, the protocols, and everything that is used with the animals, and being sure that there are some tools that are able to capture all those data sets. Then we can move to the next step, which is harmonizing, making sense of this data, and trying to see if those are linked with biological and biological phenotypes, for example. Then it's really the comparability of the data sets on their own. So using tools like FAIR3R, for example, you're sure that your data set will be able to communicate with each other because it is made to be sovereign and federated. So we have to define and use the proper tools that allow the data set to communicate with each other, and then going towards maybe the modeling part where the models are able to communicate with different data sets. They are able to make sense and compare different metadata. And then how do we build some AI models on top of this? And the goal would be indeed to create some virtual control groups. So maybe you have enough data and enough data sets that are matching your metadata contextual environmental context. So then maybe slowly you can remove or reduce your control groups with virtual control animals. And this will be a way to reduce the total number of animals that are used. But is AI the way to go? So first of all, little advertising for the behavior forum. So if you're not aware of this, you have 1 minute to Google thebehaviorforum.org, and then you will register and you will join the conversation that we have there. It's actually, if you have any interest in animal behavior, that's the place to go. And we mentioned that we are going to do this webinar on the behavior forum, and Leo Restivo, thank you for interacting there and ask this question, what's the point of curating data upfront, since now we are all fully charged with LLMs and we can just do everything at the publication moment? Of course, that's a devil advocate question, and we all know, I hope you know, that you should not upload all your research now to ChatGPT and just extract and publish what it answers. And we have to be able to still build the metadata and the proper data that will build the models that you will have to use in the future. So of course, LLMs can be a great help to understand, to do the first steps of what are the most important metadata in your research pipeline. But then the goal would be to standardize the way the metadata is gathered and then potentially build new AI models that are specifically trained on this type of data schema. And this is what you will use in the future. Also now, if you're just giving plenty of text data, that's the problem of AI, garbage in, garbage out. So you have to maybe filter what you're giving to the models in order to ensure the quality and reduce the bias of the answers. So the curation upfront, even in the AI world, is one of the key aspects, and that's what we are fighting toward. So now I will describe metadata. So it will be released open source very, very soon. So as soon as you see this on LinkedIn or anywhere, please check the repo, try to create some issues, test it, and tell me what I should change. But in short, the idea of metadata, and the reason why I created it, is that I was kind of frustrated after 10 years in preclinical research to be doing everything by myself in the lab, and I had experiments, I had some nice data, but everything was siloed and in different file format, and nothing was communicating with each other. And at the end, what I wanted to do is to share my scientific knowledge in a publication, in an article. But the problem is, as I mentioned before, when someone is reading my articles that were published few years ago, it is impossible to actually find the temperature in the vivarium when I was doing some home cage monitoring. I have this data somewhere in a server, actually in a server at the CNRS, but it is not linked with my publication because I had no tool to make this link. So this is why I thought that with the technologies that we have, we can make some one-to-one interactions, and then as soon as one software is interacting with all the third parties, it can actually make sense of this data globally. So this is why I created metadata. And this is a short demo that I made this morning. So this is the latest version that I will hopefully release this week. So it is following the investigation study assay. So here, what you have to imagine is that all the data that you see in the software might come from metadata, but it might come from a third-party application like your ELN, your electronic lab notebook, your mouse management system. As soon as they are sharing some metadata and information about the ID of a cage or an animal, it can be gathered in the same platform. So this is what I'm building, and in a way here you can see that we can have different subjects. The subject will be housed in cages, and the cages will also be attached with, for example, here what I will show is the home cage monitoring space they live in. So this is the sinusoid that could be the summary of the circadian activity. The temperature in the vivarium could be Techniplast cages or open source monitoring system that the mice are living in. You can also follow the assays, the procedures that you did on your animals in a timeline format. Interestingly, if you have a temperature sensor in your vivarium, this is what you can also do. You can plug directly your thermometer, your open source thermometer, which is actually literally the one I did. It's like a small Arduino that is communicating with the software, and you can see live the data in your animal facility in relation to actually its behavioral data set. In short, what I show here is that it does not take a lot to combine data. As soon as you have like a bit of equipment in the lab, they might have an API in the software. If there is an API, you can plug it to metadata. That's as simple as it is. Then we can also have like the facility management system. There is also a calendar if you want to organize the teams and the infrastructure. But most interestingly is like here, for example, we have 4 different home cage monitoring systems. So 2 open source one, Live Mouse Tracker, Bitbox, and 2 commercial ones, Holden Labs and Techniplast. All those are fake data that I created for the demo. But what they have in common is that all those monitoring systems have animals and an estimation of the circadian activity of the cages, and they have the strain, the sex, the temperature, humidity. And now what you can do is to start comparing the data sets and see the combination of the circadian activity, and then build what I call here the target cohort versus the virtual control group, which is simply the mean of all the animals. But what I want to show is that slowly within one facility, within one lab, using heterogeneous system, you can start building a good summary and have a good understanding of what your data are based on the comparison between environmental parameters and behavioral data. Then what we can do with metadata, and that's one of the main goals, is to be connected with different software. So here we are connected with ELN, ELAB FTW, SoftMouse, FAIR3R, Preclinicet, Techniplast, DVC cages, and my sensor for activity for temperature. So I'll show the link on how we are integrated with FAIR3R, for example. So here the idea is that you could have your dataset here in FAIR3R. And we have all the information that are directly sent from FAIR3R, and we have the same printing as you have in FAIR3R. So you know that your dataset is there. So this is good. So we are not replicating, we are just sending the same data. So it's not like a double entry. You can sort the different projects that you had and see the different aspects. So this is interesting. And then what we can do is that we can send new data to this data set. So you could select your investigation that you have in the database, the study or the results that you just finished or came from another software, and you can push it directly to FAIR3R. So this is where we are now. We validated the communication with FAIR3R. We want to expand this with other softwares, and we will add some AI capabilities within metadata app in order to make this even better as an assistant and a research agent in your labs. So those are the resources and some links that could be interesting. We can share the slides, I'm sure, after. And thank you very much. If you have any questions, here are our emails, and we are more than happy to take any questions.